Searching for papers. Okay. Okay, 101, paper 12. Again, statements, multiple statements as combinations in the options. We have to identify the correct statement. So if you are sure about something to be correct, that should be present in the right choice. If you are sure about something to be wrong, that should not be present in the right choice. Or if it is present, it is wrong choice. Flowers of Vallisneria are colorful and produce nectar. Vallisneria is a water plant. Vallisneria is a water plant. So A is definitely wrong. Anything with A should be ruled out. Anything with A is not the right choice. 1 and 4 ruled out. C, D, E are common between 2 and 3. So if you are just sure about A, then this becomes easy for you because between 2 and 3, C, D, E are common. So you have to determine whether B is the correct statement or not. If B is correct statement, then 2 is the right choice. If B is not correct statement, 3 is the right choice. So C, D, E you don't even have to look at. Just for now we can look at it. The flowers of water lily are not pollinated by water. Is it right? Not pollinated by water? Water lily pollination is? Water lily pollination? Huh? Insect and? So B is correct statement. That means statement option 2 is the right choice. C, D, E, let's look at it. In most water pollinated species, the pollen grains are protected from wetting. That is also correct. What is the coating that they have? What coating is present around the pollen which prevents them from wetting? What is it? What is the waxy coating layer around the pollen grain? Huh? Come on. What is the waxy coat around the pollen grain called? It starts with the name pollen only. Pollen kit. Pollen kit is the waxy substance present around the pollen, which protects the pollen from getting wet or getting desiccated. Prevents desiccation and wetting. Okay. Alright. The type of conservation in which threatened species are removed from their natural habitat and placed in a protected environment is called semi-conservative method, sustainable development, in situ conservation, biodiversity conservation. What do you call it? Conservation in which you take out the threatened species and place it in a protected environment. It is ex situ. Is it given in situ in the paper also? Huh?
In situ is given as an option.
In situ is given as an option, but ex situ is not given. So what do we choose? This is definitely not in situ. What do we choose? We should choose biodiversity conservation. Why? The logic to justify that is, if you let the threatened species to be in the natural habitat, they will get extinct. They will become extinct. So you are conserving that thereby contributing to the conservation of biodiversity. In situ is not the right choice there. Inhibition of succinic dehydrogenase enzyme by malonate is an example of. Succinic dehydrogenase is present where? Succinic dehydrogenase is an enzyme complex. Where do you find it? Huh? Not Krebs cycle, in the electron transport chain. In the electron transport chain. So succinic dehydrogenase is found in Krebs cycle also. Malonate inhibits it. Malonate inhibits succinic dehydrogenase enzyme. What kind of inhibition is that? Competitive inhibition. What is the characteristic in competitive inhibition? Will the inhibitor alter the active site or the binding site of the enzyme? What does it do? What does the competitive inhibitor do? It competes with the substrate for the active site of the enzyme. Is competitive inhibition reversible? Competitive inhibition is reversible. How do you reverse competitive inhibition? By increasing the substrate concentration. Okay. What is an allosteric site? What is an allosteric site for an enzyme? Allosteric site. Non-competitive inhibition, it happens by binding of the inhibitor to the allosteric site of the enzyme. What is an allosteric site? The site other than the binding site to which the inhibitor binds, thereby altering the configuration of the enzyme, which also alters the binding site configuration, preventing the enzyme from acting. Is it reversible? It's reversible, but does it get reversed by increasing the substrate concentration? Can we reverse non-competitive inhibition by increasing the substrate concentration? Not possible. Okay. Identify the part of the seed from the given figure which is destined to form the root when the seed germinates. Oh my god. This is not seed which will germinate. This is roasted seed. You don't know? Okay. By the position of the arrow marks we have to identify, no? A, B, C, D. Looks like it looks like a monocot seed, maize seed, because there is one thin unroasted portion in the center, which appears like scutellum. So probably D, no? Huh? C? Okay. Radical. Okay, just recall the parts, or recall the structures that you will find in a seed. Just recollect the structures that you will find in a seed. Coleoptile, coelurhiza, then plumule radical, cotyledons. Seed coats, tell me the seed coats. Testa, tegmen. Testa, tegmen. Which one is the harder seed coat? Testa. Okay. Bulliform cells are responsible for, don't look at the options. Bulliform cells are found in what? Are found in dash. Monocot leaves. Bulliform cells help in the conservation of water. What do? When there is no water, they become flaccid. Flaccidity of the bulliform cells make the leaves curl inwards, so preventing excess water loss. So bulliform cells are responsible for increased photosynthesis in monocots, providing large spaces for storage of sugars, inward curling of leaves in monocots, and protecting the plant from salt stress. Inward curling. Which of the following are required for the dark reaction of photosynthesis? If the question was, dark reaction of photosynthesis can happen in the absence of the same options. Light, chlorophyll, carbon dioxide, ATP, NADPH. Dark reaction of photosynthesis can happen in the absence of same options. None of these, huh? So you need light for dark reaction to happen. So coupled with the light reaction, dark reaction happens as a coupled reaction of light reaction. The products of this is used in dark reaction or light independent reaction. So that is required. Chlorophyll. Is chlorophyll necessary for dark reaction to happen? None of these is not an option. You have to choose one. So what do you choose? If you have to choose one, what do you choose? It is between light and chlorophyll that you have to choose. It is between light and chlorophyll you have to choose. Chlorophyll is a pigment. See, understand this. Chlorophyll is a pigment. Is chlorophyll the only photosynthetic pigment in plants? Is chlorophyll the only photosynthetic pigment in plants? I know the confusion that you are having between light and chlorophyll. In the absence of light, there is no ATP and NADPH production. So dark reaction cannot happen. In the absence of chlorophyll also, light reaction cannot happen is what you are thinking. No? But can light reaction happen in the absence of chlorophyll? Can light reaction happen in the absence of chlorophyll? Why can't it happen? There are other pigments also, no? Chlorophyll is not the only pigment. Suppose for some reason chlorophyll becomes unavailable for the plants. Will the plant die because of no photosynthesis? The plant will still survive because there are other pigments. The wavelength of light that the other pigments can harness is different. Correct? But they will still harness light at their respective wavelengths and the efficiency will be low, but it will still be there. Anyways, in this, which of the following are required for dark reaction? Except light and chlorophyll, all the other three are required. Carbon dioxide, ATP and NADPH. Formation of interfascicular cambium from fully developed parenchyma cells is an example of dedifferentiation, maturation, differentiation, redifferentiation. Dedifferentiation. Why? Dedifferentiation is permanent cells becoming meristematic cells. Dedifferentiated cells again becoming permanent cells is called redifferentiation. Redifferentiation. If the same four options are given and I tell you two of them mean the same, which of the two among the given options mean more or less the same? Or you can say they are equivalent terms. Which two of the given four options are equivalent terms? Differentiation and redifferentiation. Okay. You can still say because both of them are meristematic cells to permanent cells. Okay, among the given four, you have to pick the odd one out. Pick the odd one among the given four. Huh? Maturation. Huh? Why? What is maturation in plants? In the context of plants, what is maturation? Maturation is the plant cells stop dividing and start assuming functions. That is maturation. What is differentiation? It's the same. So the odd one among the given four would be de-differentiation, because the other three are references to meristematic cells becoming permanent cells. De-differentiation is the reverse of that. See the same four options. Same four options, you can have four questions there. Hint 2, Haemophilus influenza D2 always cuts DNA molecules at a particular point called recognition sequence, and it consists of how many base pairs? 6 base pairs. Tropical regions, again a logical question. Tropical regions show the greatest level of species richness because tropical latitudes have remained relatively undisturbed for millions of years, allowing more time for species diversification. See, these are again statements. Yes or no, you should be able to tell. Is statement A correct? Yes, because it has got a lot of time for species diversification to happen. So A should be a part of the option, and all four options have A. Tropical environments are more seasonal. Is it correct? Tropical environments are more seasonal. Is it right? No, tropical environments are more stable. There are not much variations in seasons. So B is wrong. Anything with B should be ruled out, and that gives you the right choice. Which one of the following is not a correct? We'll look at the other statements also. More solar energy is available in the tropics. That is correct. Constant environments promote niche specialization. That is also correct. Tropical environments are constant and predictable. See, D and E, do they appear similar? Does D and E appear similar? Constant environments promote niche specialization. Why? How does constant environments promote niche specialization? What is niche? Niche is the interaction or everything that an organism does in its surroundings. Now, if the surroundings keep changing, will you be able to define a profile for an organism? Will you be able to define an environmental interaction profile for an organism? Is it possible? No, because every day it's very frequently the environment is changing. You cannot define. So stability is something that will lead to niche development. When you do business, have you seen any businessman who says that I do everything? Ambani can say that. Adani can say that. I do everything, but they have different entities for everything. Tanishq is a brand of Tata. You know that Tanishq is a jewelry brand of Tata. Tata also has automobile business. Will you get cars in Tanishq showroom? Or because it is anyways Tata only, can you go and ask for jewelry in Tata cars? No. Or can you ask for salt, Tata salt? No, you can't do that. No? Everything has to be defined by a specific profile. If you don't have a profile for yourself, then there is no clarity about what you can be utilized for. That is niche. You should always define your niche. You might be doing multiple things, but within that, for each of the things, you should have a clear-cut profile. You might be having 10 different businesses, but each business should be defined properly. That is niche specialization. And niche specialization is achieved by stability of the interaction, stability of interactions. So that is a correct thing. Tropical environments are constant and predictable. D, statement D is talking about constancy of the environment. Statement E is also talking about constancy of the environment and predictability of the environment. Now A, C, D and E also E also is correct only. Yes or no? But we don't have a choice. We don't have an option which considers E also as a part of the right choice. Which one of the following is not a criteria for classification of fungi? Mode of spore formation, fruiting body, morphology of mycelium, mode of nutrition. Which is not a criteria for classification of fungi? Nutrition. All fungi are saprotrophic. All fungi are saprotrophic. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in the Calvin cycle? For one glucose, how many ATP and NADPH? 18 ATPs and 12 NADPH. So for every carbon fixed, it is divided by 6. 3 ATP and 2 NADPH. These are regarded as major causes of biodiversity loss. What is that? What do we call that? Quartet. Evil quartet. What are the evil? What are the components of the evil quartet? Overexploitation, habitat fragmentation, habitat loss, then co-extinction, and what else? Alien species invasion. So invasion, co-extinction, habitat loss, and overexploitation. These are the 4 evil quartet. Evil quartet. With environmental degradation and environmental loss, you remember these 4. Based on the questions, you can consider it. These are regarded as major causes of biodiversity loss. Which is not to be considered? Mutation should not be considered. So C should be eliminated. Anything with C should be ruled out. 2 options are there. Can we consider migration as a cause of biodiversity loss? We should not. C and D should be ruled out. Anything with C and D should be ruled out. We are left with only option 2. The capacity to generate a whole plant from any cell of the plant is called totipotency. Differentiation we know, somatic hybridization also we know. Somatic hybridization is done by taking which cells? Any of the cells, any somatic cells can be taken. Micropropagation. Micropropagation is used for quickly producing large number of plants. Quickly producing large number of plants. What do we use? What do you call that tissue that you use for micropropagation? Huh? You call that as a stalk. You trigger the stalk. What do you trigger in the stalk to develop callus? What do you trigger in the stalk to develop callus? You will trigger dedifferentiation. You will trigger dedifferentiation so that the permanent cells of the stalk that you have taken becomes meristematic. Callus is a mass of meristematic tissue. Can we continue to use the same stalk for a long period of time? Can we continue to produce callus from the same tissue? More and more callus? Is it possible? Why not? Dedifferentiated cells are multiplying only. No, meristematic cells, they are multiplying. You can take out that callus and you can culture callus. So callus culture is done. From the same thing you can take callus and do multiple cultures of callus and develop the plant. What are the hormones that are required for shoot and root differentiation in micropropagation? Auxin and cytokinin. More auxin than cytokinin will lead to the development of roots. More cytokinin than auxin will lead to the development of shoot. After these saplings are formed, what should you take them through before you put them on the field? What is that process called? If a question is there with that term as an option, all of you will answer that. You know what it is,
Issue of-
but you don't know what it is called, or you have forgotten what it is called. Field hardening. Field hardening. You are making them stronger for the field. See, at the end of the day, every organism has to survive in the open world. So that organism which has been trained or which has been conditioned enough to survive better in the real world is the one which will survive better. Okay. I am writing an article about how the current education system is anti-evolution. Anti-evolution. Why are we having more? We are having more graduates. When I interview people for the jobs that are available in my organization, I'll find a lot of people who are graduates with good marks on their marks card, but dumb. When I say dumb, I don't mean they can't speak or anything. The dumbness is in the inability to tackle challenges by adapting to the demands of the situation. They cannot adapt to the demands of the situation and come up with novel solutions for solving the problem. Why is this happening? They are educated, no? Why is this happening then? And one thing I am sure is, none of you will be dumb graduates, because you have been put on an evolutionary path. What is happening? What drives evolution? What drives evolution? Why, when is an organism forced to evolve? When there is a survival threat. When there is a survival threat. So, constantly pushing the boundary of comfort, intellectually also, primarily intellectually, is necessary to be able to adapt to the intellectual challenges. And most of the challenges that you face are intellectual challenges. Anything that is given ready-made and anything operated on the basis of, somebody prepares it and you use it. Somebody is preparing something and you are using it. Ultimately, using it will give you the outcome on the short-term challenge. Exam is the challenge. You are given notes by the teacher, question and answers. You memorize that. You know the questions also, you know the answers also, and the questions come in the examination. You recall by memory, write the answers, you get marks. But the exam was testing your ability to put together thoughts and respond appropriately to a question which is the challenge. So, artificial intelligence, virtual reality, augmented reality coming into education makes it more exciting. But it leads to the development of dumb people who think they are intelligent. Problem. That is why don't use any tool before you are capable of doing the task without the tool. Don't use a calculator for calculation until you can do all calculations in your head. That is why calculators were not allowed, no? Till 12th standard calculators are not allowed. In exams, we used to use the logarithmic tables. Anyways, logistic growth curve will give you a sigmoid curve. Where do you find a bell-shaped curve? Have you come across any bell-shaped curve in any of the biological concepts? Selection. Natural selection. The representation of populations in terms of the characteristics, the range of expression of characteristics. If you say height of human beings plotted on a curve, what would be the curve? Height of human beings versus the number of people in that height, with that height. So very short people will be, let's say x-axis is the height, y-axis is the number of people. Very short people will be lesser in number, very tall people also will be lesser in number. Most of the people will be in the middle of the curve. So that is what is the representation. No, it will be a bell-shaped curve. If the bell is shifting to the right, what kind of selection that is? If the bell is shifting to the right, what kind of selection it is? What kind of selection it is when the bell shifts to the right or is pulled to the left? It is directional selection. In directional selection, what is selected? One of the extremes. One of the extremes is selected. If both ends of the curve becomes very small, what is it? Stabilizing selection. If the center is disrupted and you get two bells, then it is disruptive selection, where the mean is eliminated and the extremes are selected. Okay. Spindle fibers attached to kinetochores of chromosomes during which phase? Anaphase, telophase, prophase, metaphase. Metaphase. Spindle fibers come from what? What is the organelle that produces spindle fibers? Centrioles. Centriole, centromere, kinetochore. Three terms which can confuse. Centriole is the organelle, centromere is the junction of attachment of the chromatids, sister chromatids. Kinetochore is the proteinaceous covering around the centromere. Identify the type of flowers based on the position of calyx, corolla and androecium with respect to the ovary from the given figures. See, this is a good question because they have given you two flowers. You have to identify the type of flowers. They have not given you a list of 20 flowers and asked how many of them are what type of flowers. This is conceptual question. Now look at it. What is hypogynous, perigynous, epigynous is a reference to the position of the ovary. Ovary is hypo. That means the other verbs are above it. So A is what type of flower it is? A is epigynous. Sir. A is perigynous, is it? Is A perigynous? Are all the words at the same level? A? Where is it at the same level? Calyx, corolla, androecium are all above the ovary, no? In A. Is it not? Both are perigynous. Huh? That is the diagram given in the textbook. What is the answer key? Both are perigynous. Okay. But to me it looks like epigynous, hypogynous and epigynous. Hypogynous and perigynous. The second one can be perigynous, no problem. But the first one is clearly looks like epigynous. Where is the differentiation for calyx, corolla and androecium? Adjacent to the ovary. Okay. That is how it is given in the textbook, then we have to look at it that way. Matching the questions. Match. Rhizopus, Stylago, Piccinia, Agaricus. Which one do you want to catch hold of quickly? Agaricus is mushroom, D1. Oh good. Two options eliminated. Rhizopus is bread mold. So A should be 3, A3, D1. Option 3. Stylago is smut fungus. Piccinia is rust fungus. Agaricus is edible mushroom. Piccinia causes rust disease in wheat. Stylago, fungus that affects maize, cereals. In a plant, black seed color is dominant over white seed color. To determine the genotype of the black seed plant, with which genotype will you cross it? To determine whether a particular plant is homozygous for the dominant character or heterozygous for the dominant character, you will cross it with the homozygous recessive plant for that character. That is a test cross. What will be the result of the test cross if the dominant plant that is being tested is homozygous? What will be the phenotypic ratio of the test cross? Huh? How many of the progeny of the test cross will be recessive? 0. If the dominant character is homozygous, then the test cross will not give you any recessive expression. If it is heterozygous, then there is a 1:1 possibility of the test cross giving you the recessive character also. A pink flowered snapdragon plant was crossed with a red flowered snapdragon plant. What type of phenotype is expected in the progeny? See, snapdragon is an example of what in our books? It's an example of incomplete dominance. Pink means what? It is heterozygous. Red is homozygous dominant. Pink is heterozygous. So if a pink colored flower producing plant and a red flower plant is crossed, 75% will be, 50% will be pink and 50% will be red. Pink, red, you cross it, it will be R. If you consider this one first. That's all, no? These two will be red and these two will be pink. So 1:1 ratio, you will have both pink and red flowers. Do we get white flowers also in this? You don't. Matching again. 2 or more alternative forms of a gene. Which one do you want to catch hold of quickly? See, crossing of F1 progeny with a homozygous recessive parent is test cross. Crossing of F1 progeny with any of the parents is back cross. Number of chromosome sets in a plant is ploidy. So this is straightforward. 2 or more alternative forms of a gene, allele. A3. Only one option, that is A3, B4, test cross, C1, F1 progeny with any of the parents is back crossed, number of chromosomes in a plant, sets in a plant is ploidy. You should be very careful. Number of chromosome sets in any organism is ploidy, not the number of chromosomes. Okay? Not the number of chromosomes. Lecithin, a small molecular weight organic compound found in living tissue, is an example of. Lecithin is a phospholipid. Where do you find lecithin in the cells in the body? Lecithin is a part of the cell membrane. Lecithin is a part of the cell membrane, phospholipid. Again matching. Oh, this had to come, no? This question had to come. This is the only format of question that can come. Clostridium butylicum, butyric acid, Saccharomyces cerevisiae, ethanol, trichoderma, polysporum, cyclosporin, streptococcus, streptokinase. So match them appropriately, we have our answer. Which of these four are commonly used by human beings? Huh? Which of the four are commonly used by human beings? Most commonly used by human beings. Saccharomyces cerevisiae. Ethanol. Correct. Now, as we are having the discussion in the class, the product of Saccharomyces cerevisiae action is in action across the country. In homes and in restaurants and in exclusive places of celebration called bars. Two, 3 days ago, I don't remember where, very recently, I was going somewhere for a meeting and I saw a bar open at 10:30 in the morning and it was busy. There were a lot of people inside. I am a doctor, I am working for 23 years now, I have been teaching since 23 years. I've not made too much money. Alright, made decently but not much. A neighbor in our village did not study well. He somehow got a bar license. Now he owns 3 bars. He keeps changing a car every 6 months. With the license to the bar, he gets the freedom to change his car. So it's a very scary situation, it's a very dangerous situation. People are getting more and more addicted. Alcohol consumption used to be a taboo when we were young. Today, alcohol consumption has become a part of social gatherings. So social drinking is something considered to be common. It's common. Who doesn't drink? Everybody drinks. If there is a party, they should have a drink also. People used to hide drinking to drink at home. Now, you build a big house, you make a mini bar in the house. What nonsense. Development. In the given figure, which component has thin outer walls and highly thickened inner walls? This is also roasted. What is this? Huh? What diagram is this? Huh? Stomata diagram. Looks like some constellation. Okay, then we can find out. Which one has thick thin outer walls and highly thickened inner walls? A, B, C, D. C, no, C. C is our guard cells. C is the guard cell. So guard cell has thicker outer walls and thin outer walls and thick inner walls. Okay. Which of the following is an example of actinomorphic flower? Actinomorphic is radial symmetry. Zygomorphic is bilateral symmetry. Datura is radially symmetrical. All the others are bilaterally symmetrical. All the others are zygomorphic. A transcription unit in DNA is defined primarily by the 3 regions in DNA, which are with respect to upstream and downstream ends. Upstream end you consider as the beginning of the gene. Downstream end is the end of the gene. What do you find at the beginning and the end as you move in a sequence? You have a promoter followed by the structural gene, and then you have the terminator segment. These are the segments on the gene. Inducer and repressors are not components of the gene. Inducer is something that will trigger the activation of the gene. There is something called transposon in option 4. What are transposons? They are jumping genes. They are jumping genes. What is the fate of a piece of DNA carrying only one gene of interest that is transferred into an alien organism? What will happen to it? Only 2 combinations are given. Let's see. Catch hold of something which you are absolutely sure of. That this is a possibility and or this is not a possibility. The piece of DNA would be able to multiply itself independently in the progeny cells of the organism. True or false? False. A piece of DNA integrated into the host cell cannot multiply independent of the host cell. So A is wrong, anything with A should be ruled out. Option 2, 3 ruled out. Okay, B. It may be integrated into the genome of the recipient. Possible? Yes. It may multiply and be inherited along with the host DNA. Possible? Yes. The alien piece of DNA is not an integrated part of the chromosome. If it is integrated into the organism's DNA, it will be a part of the chromosome also. So D is wrong. It shows the ability to replicate. That is also wrong. It will not show ability to replicate by itself. Oxygen is used by gardeners to prepare weed-free lawns. Why does oxygen not harm grass? Why does oxygen not harm grass? Oxygen does not affect mature monocotyledonous plants. Oxygen can help in cell division in grasses and promote growth. Oxygen promotes apical dominance. Oxygen promotes abscission of mature leaves only. Why does oxygen not harm grass? Oxygens are used as weedicides for broadleaf plants. Broadleaf plants. Broadleaf plants, dicots. Oxygens don't affect the mature monocots. Cofactor of the enzyme carboxypeptidase is zinc. Zinc. Flavin is found in FAD, flavin adenine dinucleotide. Heme is present in hemoglobin, and also which respiratory enzymes require, or which carriers of the electrons require heme, have heme center. Electron transport chain carriers have heme center. Which of these are having heme center? Correct. Below. Oh, did I hear something without anybody speaking anything? Cytochromes. Cytochromes have a heme center. Cytochromes are iron containing compounds. Zinc, magnesium, iron, these are all cofactors. Coenzymes are organic molecules. Metal ions are cofactors. Organic molecules are coenzymes like NAD, FAD, coenzyme A. Lactose present in the growth medium of bacteria is transported to the cell by the action of permease, polymerase, beta-galactosidase, acetylase. What will facilitate the entry of lactose from the medium into the bacterial cell? Permease. Permease will increase the permeability of lactose into the cell. That is the role of permease. Beta-galactosidase will break down galactose. Acetylase will combine, trans-acetylase. It's a modification enzyme. Which of the following can be explained on the basis of Mendel's law of dominance? Out of one pair of factors, one is dominant and the other is recessive. Yes or no? Can we explain that? Yes. Alleles do not show any expression and both characters appear in F2 generation. Alleles do not show any expression and both characters appear in F2 generation. That is wrong. Alleles will show their character whenever they are present. And in every generation you will see the expression of alleles based on their interaction. B is wrong. Anything with B should be ruled out. That means option 1, 2, 3 ruled out. Option 4 is right, A, C, D, E. Factors occur in pairs in normal diploid plants, correct? The discrete unit controlling a particular character is called a factor. In modern generic terms, what do we call this factor? Gene. The expression of only one of the parental characters is found in the monohybrid cross. Correct. The expression of only one of the parental characters is found in the monohybrid cross because we are studying only one character there in monohybrid cross. The statement is not correct. The monohybrid cross is a particular way of looking at things. We are looking at only one character. But do we not see the expression of parental character or other parental characters in the plant? And the cross is happening. If you look at it, you will see that also. So the statement mentioning appearing light, you will not find any other expression in a monohybrid cross other than one character that you are studying is wrong. Statement should have been better, but anyways, we will understand them. Again, statements. Bt toxins are insect group specific and coded by a gene crime. Is it right? Are they specific to insects? Yes. Are they coded by gene cry? What is the cry gene? Crystalline protein gene. Bt toxin exists as an inactive protoxin in Bacillus thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into an active form due to the acidic pH of the insect gut. True or false? False. What is wrong about it? It is not the acidic medium. It is the alkaline medium. Alkaline medium. Statement 1 is true, but statement 2 is false. Again, two statements, true or false. Parenchyma is living, but cholenchyma is dead tissue. False. What is wrong about it? Cholenchyma is also living tissue. So statement 1 is wrong. Gymnosperms lack xylem vessels, but the presence of xylem vessels is a characteristic of angiosperms. That is true. What else do gymnosperms lack? Xylem vessels in the xylem. In phloem? Sieve? What do they lack? Do they have companion cells? Gymnosperms don't have companion cells also in phloem. Again, statements continues. Chromosomes become gradually visible under a light microscope during the leptotene stage. True or false? True or false? False. When do you start seeing chromosomes? Condensation of chromosomes begin in which phase? Now tell me the sequence of. Now, if they are talking about leptotene, then it means meiosis only. Tell me the 5 stages of prophase of meiosis. Leptotene, zygotene, pachytene, diplotene, diakinesis. So when do we start seeing the condensation of chromosomes? Do we see that throughout the prophase, or do we see that in the later stage of prophase as it moves into metaphase? The later stage of prophase is when you start seeing chromosomes. Okay, not in the early phase. So you can consider the crossing over and all is over, then you move on to that. So leptotene stage statement is wrong. The beginning of the diplotene stage is recognized by the dissolution of the synaptonemal complex. Is it right? Synaptonemal complex starts dissolving. Okay. See, in prophase 1, chromosomes are formed. In meiosis, chromosomes are formed and the pairing starts. There is crossing over should happen, no? So leptotin we will start seeing. In the early phase only we will start seeing. Both are correct. Both statements are correct here. In leptotin, chromosomes are condensing. In zygotin. Okay, tell me what do you see? Leptotin, what do you see? What do you see? Bivalent. In pachitin, tetrad. In zygotin, tetrad. In pachitin, you will find crossing over. Synaptonemal complexes you will find. Then in diplotin, separation begins, chiasma. In diakinesis, complete separation. Matching 134. Nucleolus, centriole, leucoplast, Golgi apparatus. Which one do you want to catch hold of? Nucleolus. What is this? What is the right match? Site for active ribosomal RNA synthesis. A3. Two options are there. Centriole. Cartwheel, organization like the cartwheel. So B2, A3, B2. Option 3. Leucoplasts are for storage of nutrients. Golgi apparatus, site of formation of glycolipid. Leucoplasts don't have let's say leucoplasts cannot produce pigments. True or false? All the three plastids, chloroplasts, chromoplasts and leucoplasts, all three plastids are interconvertible. A leucoplast can become a chloroplast, chloroplast can become leucoplast, either of them can become chromoplast, chromoplast can become either of the others. Alright?